interrupted genes. During 1970, in some mammalian virus, for example adenovirus, it was found that the DNA sequences coding for a polypeptide were continuously but were split on into several pieces. Therefore, these genes were previously termed as split genes or introns. Interrupted genes are intervening sequence inserts or junk DNA. For the discovery of split genes in adenovirus and higher organisms, Richard J. Roberts and Philip Sharp were awarded the Nobel Prize. Split genes were independently discovered by Richard J. Roberts and Philip A. Sharp in 1977, for which they shared the 1993 Nobel Prize in Physiology or Medicine. The first observers of interrupted split genes, the genes in which there are non-coding intron sequences between the coding exon sequences, were made in animal viruses in 1977. Introns and exons. Split genes appears to be complex and fragmented genes split up into alternating coding and non-coding regions. The non-coding regions are collectively called junk DNA, functionless as DNA. The coding region containing actual information of the genes are interrupted by few to several non-coding sequences called introns from intervening sequence, which are spliced out after transcription. Such genes are called split genes since their coding sequences are split into several parts due to introns. So interrupted genes, exon, intron, exon portions, transcription, primary mRNA, then splicing will be happening. So mature mRNA is formed. Introns and split genes are very rare or absent in prokaryotic, mitochondrial and chloroplast DNA, but relatively numerous in most eukaryotic nuclear genome. All the exons undergo transcription and translation, so they find their expression in the primary, intermediate and final product, pre-mRNA, mature mRNA and protein. Introns are only transcribed to the pre-mRNA and sliced out during post-transcriptional chemical processing of immature mRNA. So gene, exon, intron portions are like this. Promoter is also there. So transcription, RNA. Later splicing will be taking place. So mRNA contain only exon portions. That amino acid gene, post-translational modification, later that protein is produced. Then mRNA splicing. For most eukaryotic genes and some prokaryotic ones, the initial RNA that is transcribed from a gene's DNA template must be processed before it becomes a mature messenger RNA or mRNA that can direct the synthesis of protein. One of the steps in this process, called RNA splicing, involves the removal or splicing out of certain sequences referred to as intervening sequence or introns. The final mRNA thus consists of the remaining sequence called exons, which are connected to one another through the splicing process. Nuclear pre-mRNA splitting is catalyzed by the spliceosome, a multi-megadalton ribonucleoprotein complex. Both the conformation and composition of the spliceosome are highly dynamic, affording the splicing machinery its accuracy and all the same time flexibility. The mechanism of mRNA synthesis for a DNA containing introns can be explained as RNA polymerase may synthesize both introns and exons, and processing of transcripts occur later on. The transcripts corresponding to introns are removed. The first reports of split genes were provided by exon gene of adenovirus. The DNA of adenovirus is double-stranded and codes for 20 proteins. 8 out of the 20 genes are called late genes because they transcribe mRNA late in the virus life. The 8 late genes transcribe as single RNA, which is later transformed into 8 mRNAs. The total length of the 8 mRNA is, however, less than the length of the original transcript. When one of the genes, heron genes, was paired with its mRNA, the DNA formed 3 loops which represent the unpaired region. This indicates that the heron mRNA was transcribed by 4 different regions, unlooped regions, and the 4 segments were then joined together. The looping is means to exclude unwanted DNA which does not transcribe mRNA or introns. So exon gene of adenovirus with its transcribed mRNA. A, B and C represent regions of DNA which do not transcribe DNA. Then evolution of split genes. Before the discovery of split genes in 1977, all the genes analyzed in detail were the bacterial genes. Bacteria were considered to resemble with the simpler cell from which eukaryotes must have been evolved. Now it is supposed that split genes are the ancient condition and bacteria lost their introns only after evolution of most of their proteins. Evidence for the ancient origin of introns has been obtained by the examination of the genes that encode the ubiquitous enzyme triose phosphate isomerase. The triose phosphate isomerase is coded by a gene that contains 6 introns in vertebrates. 5 of these are present at the same position in maize. This shows that 5 introns were present in the gene before evolution of eukaryotes about 109 years ago. The TPI or triose phosphate isomerase plays a key role in cell metabolism that catalyzes the interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, a central step in glycolysis and glycogenesis. By comparing this enzyme in various organisms, it appears that the TPI evolved before the divergence of prokaryotes and eukaryotes form a common ancestor cell pre-genote. Pre-genote intron exon E. coli, Saccharomyces cerevisiae, Aspergillus, maize vertebrates billions years ago, 1, 2, 3, 4, like that. The unicellular organisms under strong selection pressure minimized the superfluous genome in their cell, whereas there is no such pressure on multicellular organisms. This is why Aspergillus has 5 introns and Saccharomyces has none. Precise loss of introns would have occurred by deletion in prokaryotes. The loss of introns require the exact joining of DNA coding sequence outlining of evolution of a particular gene. The most likely source of the information is needed for such event in an mRNA transcript of the original gene from which introns are to be removed. The mRNA may be copied back into DNA by a reverse transcriptase. Then feature of split genes. The organization of split genes would be consistently the same in all tissues of an organism. Introns all by themselves don't have any coding function, but exon and intron combinations code for mecturase protein encoded by exon and intron combination. They catalyze the excision of introns from the primary transcript of introns for the primary transcript and subsequent splicing of exons. Exons have precisely the same order in the split gene and also in its primary and secondary mRNA transcripts. The presence of introns is neither essential nor deleterious to the organisms. All the split genes are begin with exons and end with exon. Primary mRNA transcribed from a split gene known as heterogeneous nuclear RNA, hnRNA, or precursor mRNA. It might be exact replica of the split gene with alternating exons and introns.